So first of all, this is the exercise. I mean, Sadie, have you ever the chance to try it?
Yes, I had success all the way through almost the end. I think in the very, I can't see it right here, but I had some challenges downloading some of the other data. I followed the instructions, but I don't know if there was just something going on with.
Well, that would be nice to
or
check.
or I was picking the wrong things. But it didn't seem to take, I mean, it seemed to upload the data, but then when I went to do the evaluation steps, it wasn't finding it. So either wasn't finding it or I wasn't sure where to look to find.
Maybe you can, I mean, after you can share your screen, we can have a look what's going on. Dalton, have you had the chance to run the exercise yourself? Well, I don't know. When you're back or when you're here, just let us know. So I would do it in a. So here you basically have dissected all these steps. They're not concatenated. Of course, you can do it manually, and you did for sure. But there are, like, especially if you have a pipeline already in mind, there are bit more efficient ways to do that. So I'm going to try to do it with you in such a way. So I'm going to connect to Galaxy. Sorry, connect to Galaxy as well. Of course, everybody didn't have a problem with setting up the account. As you can see, my history is empty here. And so one way to, I mentioned this last time, but one way to actually, oh no, I'm not logged in actually. Yeah. So one way to actually work with Galaxy, which is very convenient, I think, is to create a workflow. If you have to redo the same analysis multiple times, or you suspect you will have to, it's very convenient so that you don't have to remember all these steps by heart. And we call it two. And we have this sort of whiteboard where we can basically draw anything we like. So we want, first of all, to dump the reads, right? Do you have, I mean, any of you has ever had experience with the sequence re-archive? I mean, do you know what the sequence re-archive actually is? Have you ever deposited data on the sequence re-archive or retrieved data from there? Well, I guess that's a no. Okay, so we want to run fastQDump first of all. As I mentioned in the exercise, that there are actually two versions of fastQDump. There's fastQ and fasterQDump and I mean, this is exactly the same tool that you can install via Conda or directly from in CBI. That it's the interface with the sequence re-archive. FasterQ was developed a bit later to be faster. It's, in my experience, is not a very say efficient tool. So sometimes it gets stuck, it complains. But overall, I mean, it's if you, especially if you can parallelize the download, is actually not too bad. So you can drag and drop something here, and we know we want to get the data, but of course at this point we're just designing the pipeline. So the first step is running the QC, right? So we want to add a QC with fastQC. Here we go. We just need to connect. You can see that you can actually decide what to connect to, which, and of course we're going to connect with the short read data from your current history, which basically means from here.
So one thing too is when you start each application, you can also see, like in a pull-down menu, what inputs are available for this particular application.
That's right. So here on the right, you have all the options that are normally provided, even when you browse and click on them without being in the workflows area, and you can decide what to do exactly. For instance, here you can decide whether you want to keep it as a compressed file or uncompress it for some applications. And this is the same also for FastQC. So you have a bunch.
So Galaxy is smart enough to every output know the data type, and is matching data type with the input type from the other app. So you rarely make a mistake by using the wrong file in terms of data type.
It's something you may have not tried because you actually use FastQDump to get your data. But if you upload the data yourself, that's also pretty fast. And basically when you upload them, it's smart and it can recognize what kind of files most of the time you're dealing with. So then you can use it throughout the pipeline. Here, of course, with FastQC, we have the possibility. Then I would like you to share your FastQC results. But basically, you have a series of metrics representing the quality of your real data. Okay, so we're going to look into that in a moment. But let's move on to the next step. So we want to, as you can see, we can assemble it on the fly, so right away. So here, as you have noticed, we're using IFIasm, which is the fastest and well, there is always some competition there, but one of the the fastest assemblers for hi-fi reads that we mentioned on Monday. So let's say the high quality long reads from PacBio. And again, we just drop it here. Of course, at this point, we don't want to connect to the FastQC step, but we're just going to drop another connection from FastQDump. So we download the data, we can run the FastQC, and in parallel, we can already start the assembly process. We don't have to wait for the FastQC to finish. And again, here you have, let's say, more or less advanced options for IFIasm. We're not going to go through those options, but there is also usually a good explanation about the options here, or you can find that explanation on the website of the tool. Next step would be to actually collect let's say the general, what I call here general genome metrics. Here, I mean, Galaxy is not so versatile, so it doesn't contain necessarily all the tools. You can always pull a request. So you have a specific tool in mind that it will be helpful for you. You can ask the Galaxy developers to actually implement it for you. They can actually be pretty fast and efficient to that. But otherwise, you maybe have to, play with the tools that are available to get to the results that you want. So here we want to run the K-Mer counter, right? So the K-Mer counter is K-Mer, and we want the all-in-one thing here. And again, this is also another QC step, so it's basically in parallel. So it means that, again, we can write potentially at the same time as the other steps. Then remember here, we have to, if you have reached this step on your own, there is some little conversion of the file. But by the way, I have to remember that we want, in this particular case, we want to set some advanced parameters. Here, show K-Mer size. So K-Mer size, I mean, of course, between 20 and 21, there is not such a big difference. But there is a famous paper that mentioned that 21 for several species is actually a good length. For other, it really depends on the different group. So, and also the different application. But here, again, I just use 21. Then let's get the table size increase. So the table size essentially means how many counts for each K-Mer are allowed as maximum. So if you have a K-Mer that is seen millions or billions of times in the assembly, sorry, in the raw data, you may be excluded because the table size is not large enough to actually also include that high frequency count. So that's why I'm increasing it, because otherwise you would see weird results at the end of the pipeline because they get excluded. But actually here we want to get the full spectrum of K-Mers. And then we have this little step here that I don't know, if you're not familiar with AWK, it may sound very exotic. Who among you knows how to read this? It's not that complicated actually, but you need to know what AWK is. Anyone? No? Olivier, you must know that.
Sorry, repeat the question.
Come on, you're not even listening. Right? The question was about what does this particular AWK command does in this case.
Wait, can I try before, Olivier? I'm gonna butcher it, but can I try? Okay. It seems to me like you know, an if-then statement, but it's saying if you get this, and I want to say maybe it's referring to the quality score of one of your your base pairs. If it equals this, then you're having it print something else. You're giving it the command of what to sort of convert it to.
Almost. So the FS it's tell you that it's separated by commas. So you're going to split your text into with the commas. The if condition test, if we skip the first line, right? And if your value on the second column that you split with commas is different from 0, then you print column A and 2, column 1 and 2, right? So you extract the two first columns on comma separated, and you skip the first line.
Yeah.
So you're going to get the long list with 2 columns.
So I think it's useful to understand what's going on here, and it becomes obvious when we use your results that you already have to compare the file before and after we apply AWK transformation to it, okay? We can do that. So as a general terms, I mean, AWK is basically a binary file and a software that allows you to convert and manipulate any kind of file. It's very, very efficient, so it's close to compiled software. It's not, because of course the original file is a plain text file that you're trying to manipulate. But in terms of speed, is really, really close to that. That's why it's so popular. And as you can see, you can compress in very short length a lot of let's say options and things that need to happen, a lot of instructions to run. So maybe we can come back to this after when we look at your results, so you can compare before and after. But basically,
so, can I add something, Giulio?
Sure.
So this step is going to reduce your big CSV file that contains like 5 columns, I think, into 2 columns, and the 2 columns are going to be the K-Mer and the count of the K-Mer.
Yeah.
That's the genome scope.
Yeah, but again, I think it becomes clear after a while. I'm just going to add it here now so that we know that it exists. So I'm going to put the actual command in here, and I'm all set. And then we want to do this step of GFA to FASTA, so to convert the output of IFISM, which is in this kind of weird format, if you want, GFA. FASTA is, the most popular file format for sequence data ever created. back in the 70s. But GFA is actually containing more information, so it's richer in information because it's a graph file that contains information about the, let's say, structural or even single nucleotide variation that is observed in a particular sample, individual, and so forth. These are relatively new file formats. So the GFA, I think it's maybe 4 or 5 years, no more than that, but it's becoming more and more popular so that even IFIASM does not even output the FASTA by default. I think that was actually a very, a choice by the author of IFIASM, because he's also one of the developers of the GFA format, so that they basically thought, okay, so that we're going to make it more and more popular. But it makes sense because actually, again, you lose less information. The FASTA does not contain a lot of information. So here from IFIASM, we're going to have basically two GFAs here. Sorry, GFA here and here. One is the primary assembly, and the other one is the alternate. We're going to come back to say a few more things about what these two assemblies are. I mean, what's the difference between them? But here is to say that we get two assemblies out of the assembly process with IFIASM. And in a diploid organism, they correspond to the two basically haplotypes of the genome. In other cases, they may correspond to, even if it's, let's say, a haploid cell line, like in the case of yeast or the E. coli, they may correspond to some variation, like erythroplasmy or similar, that was actually observed by the tool in your sample. So we convert it, and after we have converted it, we can run this FASTA statistics that will give us some information about the two assemblies that we have for both haplotypes, right? Then we want to run Busco. Did you get to run Busco? Yeah. And again, Busco is going to run in the same files, right? So here. And you can see how let's say, complicated, but also relatively easy to handle the pipeline can become using this kind of tools. I'm now going to run the compare step here, because that's probably something that one will do only at the very end, depending on the results. But yes, this is pretty much it. Oh, there is in Busco, you've seen that there are options to set here, like E. coli should be this gamma proteobacteria. Where is it here? Otherwise, you get weird results. But we're going to talk about Busco right now. So I'm just going to launch it. We still have potentially 2.5 hours, or we could also decide to finish a bit earlier if you want, I think. So I'm going to run the E. coli as well, so that and let's see if if it did everything correctly or not. So I'm going to save or run the workflow. Hopefully it's going to save it. Yeah, so here is complaining because of course there is no accession provided, but I'm going to provide the client accession number here. Now it's not complaining anymore. And let's see what happens. Okay. Olivier, do you have comments?
Yeah, I mean, that's great to do a pipeline like that. But for the first time, I mean, if you're not comfortable building the pipeline, you could run each application independently and plug them in. and I put that in the chat thing. But once you've done all your applications and finished, you can transform this history into a workflow. So you can save it for later and to redo it with another data set. Personally, I like to do one application at a time at first so that I know what I'm doing. But yeah.
Yeah, and in fact, there is a very small chance that what I just did not work. But let's see. But what is nice though is that you see now that all the jobs were scheduled at the same time. And again, if one fails, all will fail or many will fail. But let's see what happens. So now before we go to your results, I think it's good to introduce some of the concepts that are actually relevant to, I mean, some of the things you actually run, but probably we didn't know exactly what you were running for the exercise, and some other things that are not part of the exercise, but I think it's still important to mention them for you to know. So I think Olivier and I have been looking at these plots for years now. Like,
yeah, I dream about them.
Yeah, yeah, exactly. How many of you have seen such kind of plot before? I know Sadie did, but
I haven't.
okay. Normally, these kind of plots are actually a bit scary. I mean, if you're not used to it, or if no one explain how to read such kind of plot, but it's one of the most powerful tools in the say Genome Assembly community. It was developed by Mike Schatz's lab, GenomeScope 1 a while ago now, and GenomeScope 2, which you may have run already, right? It's actually extending their range of applications to polyploid organisms as well, so not just to diploid and haploid. So here we are dealing with K-Mers, so there is always a first step, which we also run right now with the K-Mer application to count the K-Mers. So counting the K-Mers, if you remember, counting the K-Mers is doing this operation. So we have a sequence ATG, TG, TG, and then we can enumerate the K-Mers. All the K-Mers that are part of the sequence are ATG, TGT, GTG, TGT, and GTG. Okay, same for the other sequence. But actually, when you count them, it means that actually you assign a number of occurrences to each K-Mers that you see. Okay, so this is the K-Mer counting step, where you actually have 0 copies for this particular K-Mer, 1 for the yellow one, and 2 for the other 2 K-Mers in this particular sequence. And here you have to imagine that you're going to do this not just for a bunch of K-Mers like here, but you're going to do this for all the K-Mers in your row reads. Now we're talking about millions of reads, okay, of short reads in particular, but also the hi-fi reads, which we have been using. So we have a ton of K-Mers. Here we have very short K-Mers, so only 3 letters. But if you, as we did, use 21 as length, so you're computing 21 mares. Okay, of course, the number of potential K-Mers is a bit smaller. If you use let's say the shorter the length of the K-Mer, the more K-Mers you're going to get. Okay, not necessarily in terms of, actually you're going to get less potential combinations, of course, because you have less potential combinations here, but you're going to get many more counts of them. Vice versa, if you have a ton of very long K-Mers, you actually get a lot of different K-Mers, but then the counts, of course, decrease. So there is an inverse relationship here. And so why is it so important? Because actually, as I mentioned last time, K-Mers is basically enumerating all possible sequence that you observe in your raw data. Okay, you only define the length, and after that, you have basically a representation of all the sequences in your raw data. What the K-Mers are missing is the connections between them. So you lose the order. When you count them, when you have a table like this, you don't know anymore whether ATG came before TGT or not. That's where, let's say, the K-Mers fail, and that's where the De Bruijn graph actually. That's what the De Bruijn's graph are actually about. It's just telling you what is the connection between the K-Mers. Nothing more than that in essence. Okay. So the De Bruijn graph is going to give you the K-Mers and their connections as edges, and this you can use for the assembly. But we can also use the K-Mers as they are, so without having to look into the De Bruijn graph and the connections between the K-Mers. And that's what Genome Scope achieves. Okay, so we have we create all the K-Mers from all the reads. So imagine you have a 150 base pair long read, and you dissect it into all the 21 mares that constitute that read, or even a long read, high fine, that is 20 KB, and you extract all the K-Mers that make up that read. And then, of course, you pile them, if you want, into considering all the K-Mers from all the other reads. So that in the end, you grow this large table of K-Mers and counts. Okay, so K-Mer, for each 21 mare that you observe in your sequences, there are specific counts associated to them, which could be 1 or could be 1,000,000 for something that is always seen, not a very frequent sequences in the genome. So when you have these two information, you can extrapolate, and this is what happens here, and you can create a table that does not, that aggregates the counts, okay, the counts for each K-Mer by their actual observed number. So to put it differently, if you have 2 K-Mers, each one is seen 20 times, okay, you can now say, I see at the coverage of 20, I see 2 K-Mers, okay? And you can do the same for all the K-Mers that you have. So it means that you have I don't know, 1,000,000 K-Mers that are seen 20 times in the assembly. So it means that around 20x coverage, so 20 times that particular observed K-Mer, you have 1,000,000 of them, okay? And of course, you do this for all the K-Mers that you have, and you end up with this plot. So coverage means how many times you observe a particular K-Mer, okay, in the row reads, and the frequency is the sum of all these K-Mers that you're seeing in the row reads at that particular frequency. Does this make sense?
It does to me.
Well,
Yeah.
you dream about it, so it's not.
Doesn't mean I dream true. I mean, I may be wrong in my dream.
All right. So what do we have here? Genome Scope takes this profile. So the profile means that when you do this operation, what happens is that depending on the size of the genome, on the heterozygosity of the genome, on many characteristics, you obtain a spectrum like this kind of profile that is unique to that particular genome and reflects the characteristics of that genome. So it means that basically what they did with Genome Scope is say, okay, so if we have this unique spectrum, can we use it like we identify from the unique spectrum a chemical compound? Can we use this to basically predict from the K-Mers some properties, some characteristics of the genome that actually from which the data was generated, not from which the row reads were generated. And this is what happens here, and it's very, very, of course, it's error prone as anything in bioinformatics, but it's actually very, very accurate at this point. So we're getting very, very accurate information. So the first thing we can see here is that there are 2 main peaks, and there is then this big peak that is peaking somewhere we don't see in the plot. Okay, so want to say something? So the
is the size of the K-Mer varying here, or it's constant?
No, so this is always 21. But that's the
21.
only thing you have to decide up front. You can use different lengths. It doesn't change much. I mean, for some application, one could use a different number, okay? But actually, it doesn't change that much. But the point is that you have to, when you count them, you have to count them all with the same length.
Yep, makes sense. Thanks.
Okay, and so we have two peaks. These two peaks correspond in a diploid organism, as in this case. I don't even know what this is from, honestly, but in a diploid organism, so we have two copies of of all chromosomes of the genome. We have a diploid peak and an haploid peak. Okay, so these two peaks actually correspond to those sequences that, as judged by the K-Mers, are seen either essentially at twice the haploid coverage or at the haploid coverage, meaning that, of course, whenever you sequence data to assemble a genome, or even if you have a resequencing experiment and you just generate some whole genome sequencing data for a particular individual, you never, of course, generate 1x coverage. You generate 30x, 60x, 100x. The reason why you want to do that is because the sampling, let's say, of the genome is random. Okay, so you may end up sequencing 10 times one region, but only 5 times another region, just by chance, just because it's a random process of extracting basically pieces of DNA from a jar. Okay, but so you want to increase your chances to represent all the genome. So if you sequence 100x, there is, of course, something that is sequenced 50x, something that is sequenced 20x, because it's a Poisson distribution. But at least you get a good representation of everything. If you sequence a 10x, what happens is that your left tail, so the stuff that by chance has been sampled less, it's maybe 0 times, okay, because it's never got sampled. Okay, so that's why you want to increase your coverage. So here it's telling you, okay, so when I look into the K-Mers, I observe essentially the same pattern. So I observe that I've sampled the K-Mers at a certain coverage, okay? And if you have a diploid organism, what happens is that you expect that most of the genome is actually identical between the two copies. So you will have twice the number of K-Mers that correspond to a particular sequence, because the sequence is present in both the, let's say, paternal and the maternal haplotypes. Okay, so if your average coverage of the haploid genome, of a copy of the genome, is, let's say, 30x, and you have a diploid genome, you expect that all the sequences that are identical between the two copies are going to be seen at about 60x. Again, it's a distribution, so there will be something that is 2 copies, but just by chance will be sampled at 55x or at 65x. Okay, so, but overall, this is respected, and when you count them and pile them up, you end up with something like this. So a distribution that is centered around about the approximate coverage for the diploid, in this case is maybe 50x, and for the haploid, which is about half of the diploid peak, so 25x. Okay, and you can also see the GeomeScopes try to estimate these peaks. The peak here, it's complicated because you'd have to imagine that this is the result of overlapping curves. So it doesn't mean necessarily that the peak corresponds to where the estimate of the real average haploid or diploid coverage is supposed to be. That's why it's a bit shifted. But in principle, they correspond quite a lot. Okay? So basically, this is how you look into this plot. There is a, then I will ask you if you have a question, but there is another important consideration here. So there is a lot of stuff that, as we can see, is very, very high frequency, but very, very low coverage. So whatever it's high frequency and low coverage is the result of sequencing errors. So imagine that you have generated your reads. It's millions of reads, and we know that Illumina, HiFi, they all make errors, sequencing errors. So these sequencing errors are almost random, right? So if they are almost random, it means that they are not seen many, many times, okay? So imagine you're looking at a specific region and, I don't know, 10% of your reads make a mistake in that region, okay? But they don't make all the same mistake. So there are these little errors here and there. When you compute the K-Mers, what happens is that you get the majority of K-Mers right, so they correspond to the real sequence. But some of them, which overlap with the errors, will actually end up being counted, but they will have very low frequency because we never see them in all the reads. We only see them in the few reads that contain that particular error. So that's why they pile up at the beginning. And this is very interesting because we can use now this information to say, okay, all these K-Mers are errors. So if I, let's say, look into my assembly, I see one of these K-Mers, that's an error, okay? Because it's very, very low frequency or almost very likely to be an error because it's very, very low frequency in the reads, okay? So this is how basically you read this plot. Of course, there is another, this curve continues to the right. We're going to talk about this in a moment. But as you can see, the vast majority of K-Mers is centered here. In fact, most of the genome is actually unique. It's not present in a very large number of copies. But even then, when it happens, it's really a lot of copies of something. So it's not necessarily like 3 times. It's maybe 100 times. Maybe those are, I don't know, transposable elements. So stuff that maybe it's present in many, many copies. So it's not going to be seen here, but it's going to be seen if you actually go to the right of this plot for a while, and you start to see a lot of sequences is highly repetitive. So here what we're looking at is the unique fraction of the genome plus the error of sequencing. Olivier?
Yeah, I would like to emphasize this is without any assembly information. I mean, this is quite amazing, right? No alignments, no assembly information, nothing. So everything from the raw data that has been like, the complexity has been reduced to K-Mer so that you can interpret things. Yeah. So you do that before doing any assembly or any work. This will tell you how your data looks like. I don't know if Julio, you have some example where the distribution looks weird or there is something skewed that
No,
there's something wrong with the data, right?
I don't have other examples, but yeah, so there are a few things here. So first of all, you can use this information to estimate the size of the genome. So again, as Olivier was saying, we can estimate that what will be the predicted size of the genome, even in the absence of any assembly or any other information. And these estimates tend to be, not necessarily always, but tend to be very, very accurate. Okay, not at the base pair level, but in terms of megabases, they usually are. So this is a relatively small genome. Again, I don't remember what species this is, but you can see it's half a gigabase. So I don't know, it could be, I guess, a butterfly or something like that. But the other thing is that here we have 2 peaks, right? And we said that one peak, the diploid peak, is here, and there is a haploid peak. So the relative proportions of these, because these 2 peak and their height is influenced essentially by how much the genome is heterozygous. Now we said the diploid peak is all the stuff that in the genome is represented twice. Likewise, the haploid peak is whatever is present only in one of the 2 copies. So if, let's say, the mother and the father were very distantly related, they will have a highly heterozygous child, okay? And that means that the haploid peak is going to be much bigger. In this case, this is actually a very relatively heterozygous individual, okay? Normally, this peak can be very low. If you take an inbred individual from a colony in a lab, for instance, this is going to be near 0, okay? Because they are all almost genetically identical, and therefore the heterozygosity is close to nothing. But here it's almost 2% heterozygosity. So this is huge. This is a very, it's definitely from some wild, healthy, I would say, population, because as you know, the heterozygosity is also linked to basically how large the population is, and therefore also how endangered, if you want, the population is. So you get also this other estimate, and this is an important estimate in the genome assembly world, because depending on how heterozygous an individual is, you may want to use different tools to assemble its genome. Because essentially the tool can work better if, let's say, there is a very low heterozygosity. That has been the case for a very long time. In fact, for genome assembly, people tended to use highly inbred strains or species or individuals, because they actually could provide you with a single, basically, copy of the entire assembly, because there was no difference between the two copies. And this simplified a lot the overlapping, the problem of finding the overlaps between the reads. Now, there are methods that actually benefit from the opposite situation. So when you have a very heterozygous individuals, you actually can separate much better the two haplotype and assemble two full copies of the genome for both maternal and paternal haplotypes. How is it going? Questions, doubts? Anyone willing to repeat this explanation?
I find this really interesting. But I was just wondering, with
So
this.
Matthew, right? You're still with us. Okay.
I'm still with you. I'm still with you. I'm paying attention. So the haploid versus diploid peaks, you can't actually. Can you infer based on the ratio, like the actual proportions of haploid versus diploid uniqueness, or because of all the stuff over on the right, is that going to really pollute your ratio?
No, so the way GeneScope works is that it's going to use it's an R function to fit a model of the most probable underlying distributions that provide this kind of situation. So it means that it's going to try to fit a distribution for the diploid stuff, okay? One distribution for the haploid stuff, and it's even going to fit a distribution down here for the 3-copy and the 4-copy stuff. Okay, so it's trying to iteratively fit this distribution. It's actually very fast if you have tried. And when you get to the point that it's fit, you have estimated the relevant parameters of the distributions, and then you can make some very fancy computations. So it's really, I mean, it's really sophisticated in that you have a lot of basically, it's simple in a way, but it becomes, the math becomes very, very complicated quite rapidly, because you have to estimate all potential combination of alleles that could contribute to this peak or to this other peak. So you have like relatively large mathematical equations in the GeneScope model that, taken these model fit, generate your expectations about the actual heterozygosity given this particular distribution. Does that answer the question?
Yeah. And so if you go up to like something that's like a tetraploid or beyond, is it just peaky all the way down?
So I think Michael Schutz told me at some point that the GeneScope 2, which is supporting aneuploidy in principle. Of course, the more diploidy, the more this becomes confused. So at some point, it doesn't really work. But the thing that they're fitting like 200,000 curves, so, sort of, sorry, not curves, but the underlying models. They're trying to compute all the, and there is basically a loop and a series of loops that create all these complex formula that then the software tries to solve. So yeah, I mean that was actually, even if you just go to tetraploid. So if you imagine to go beyond that, it's a nightmare. Yeah, and that's why, by the way, I mean most of the good quality genomes are actually for vertebrates, or at least diploid species, not for plants. Okay, because plants, it's I mean, some plants are actually easy, but some plants are extremely complicated.
It also depends when the polyploidy happened, right? So if it happened recently, you may have a bigger diploid peak, a gigantic diploid peak.
Yeah.
Yeah.
Cool.
Like, yeah, if you have a complete duplications of the genome and you don't allow too much time for evolution there, maybe you're still okay. But otherwise, it becomes complicated. So here to the right, you have basically what happens when you start to go beyond 120. Okay. And you can see, so what happened here is that the axes were log scaled. Okay. So you have these sort of weird compressions of the curves that we observe here. Now the one that is going down rapidly here goes up because we're close to zero in log scale. And then these two, I mean, the difference between these two becomes less pronounced. And of course, I mean, in this case, it's not really good to estimate the parameters. Well, that's not what this plot is for. But actually, this plot is, and sorry, you can also see the peaks that correspond to these 4 lines here that are replicated here. But actually, what this is actually very useful for is to get a sense of what's going on in terms of high frequency K-Mers, so the repetitive fraction of the genome. So here it means that, okay, this is going down very fast, but if you compress it and pile it up, you see that there is still a lot of stuff that is not unique, so it's repetitive. Could be just 6 copies, or it could be millions of copies, okay? Here we're talking about 1 million copies. So actually this is useful because as you can see, there is also an estimate here about the fraction of the genome that is unique versus the repetitive fraction of the genome. So we're talking about, in this case, 70%. And again, complex formula, but the idea is that you take whatever it's under the black curve, this is your unique fraction. Whatever is left to the right, it's your repeat fraction. Okay, and you can use this information to ask. Of course, it's not just, you know, what it looks like here, because this is also in log scale, but it's pretty much the idea. And again, here, if you have a very repetitive genome, in this case, it's not particularly repetitive. We're just talking about 30% repeats. It's going to look much different. Okay. Okay, so did anyone have the chance to run this on E. coli or some other sample? Want to share your screen and show us? Can stop sharing. Did you or not? I'm going to stop sharing my screen anyway. If anyone can. By the way, my FastQC in the meantime, it's done, so I was able to download the data, but we're still running the assembly.
So I'm showing my screen. I think this is it.
Yeah, so yeah, that's good. That's very good. So here you have the table of the counts. So you have the first column that is the coverage that we were talking about. The x-axis in the genome scope plot is exactly the same information, and then you have on the y-axis you have the second column. Okay, and you remember that we've seen that errors have a very tall peak that we didn't even see, and you can see it here. Now there is 22 million errors. This is the E. coli, right?
This was the first one. This was yeah, E. coli. This was E. coli. Yeah.
So, and then the third column here is just the sum of the previous column, so the cumulative sum, and the last one is the relative fraction. But we're not interested in these 2 final columns, and also we don't, of course, care about the first. The 000 stuff. That's why we use AWK to actually remove it. If you scroll down, we can actually start to see what's going on at the high frequency. So you see that it's dropping now. That when we get to 100, we now have 1 or 2 copies. Now there are very few K-Mers at high frequency, 1, 2, and so forth. And you keep scrolling down, you can see that this is, well, in this case now it's going up again. You can guess why, because this depends. Basically, we went down, then we went up for the peak, and then down again, and then there is again repetitive stuff, which is low frequency. You see, very few copies, but this table is massive. You know, it goes on and on for hundreds of thousands.
That's just E. coli. That's a simple case.
Yes, and there's a very simple genome, no? Because we're talking about what is it, 5 million base pair, but the genome that was actually relatively small that I showed before, it's 100 times that size. Okay, and of course this is gonna go on and on. Okay, so have you run also the AWK command?
Say that again?
Have you run the AWK command as well that converts this file? That's reformatted.
Text reformatted. Yeah.
Yeah.
Okay, so that's good. So you see the difference now. We have columns. So
This is that step here, right? This was this thing.
Exactly. So it's just a very simple trick to convert a tabular file into another tabular file. And here we just did two things. We dropped 2 columns, the last 2 columns, as I said, we did not need them. And we also removed the 0, 0, 0, the first line that is not really necessary, okay? So this allows us to run genome scope. Have you tried that?
Also, we remove the first of the header.
Yes. Oh, and we removed something more than that. That now I'm going to explain how you read the AWK again. But we also removed all the coverage values that were at 0 frequency. Now, when we go to the high repeat stuff, the highly repetitive stuff, yeah, if you scroll down a lot, you see that there are many 0 values here, like yeah, all of them are actually 0. And of course, this is not really necessary for genome scope to work. So we get rid of them, and this is what you actually got in here. So if you scroll down, now you will see that you're actually skipping some of these values because of course they were 0 and we don't actually need them. Just as a curiosity again, if you want to understand the command of AWK, can you go to that page again? Say it, please.
Go to this or the first one.
The AWK or the exercises. Yeah. So here, if you remember how the first file is formatted, we have a comma separated rows. Okay. So FS tells AWK consider the comma as a separator for columns. So the columns are going to be separated by column. And it's, I don't know, if you play with Excel a lot, for instance, you do that all the time. Now that when you import the file, maybe you have to tell Excel which kind of file, which separator you have. So here is the same thing, same principle. And then we have just the 2 very simple conditions. The first one, if the second column is equal to 0, sorry, if the second column is different from 0. So $2 stands for second column, and then what's called this equal sign, and then 0. So if it's different from 0, then print the row. Because the way AWK works is that it takes the file and goes line by line and applies your rules to each line. Okay, so here it says if the row I'm looking at, its second column is equal to 0, skip it. I'm not going to print it. Okay, and then of course we're also skipping the very first row because it's the header. And you can see that you can then use AWK with printf, which again, if you're familiar a bit with the command line, it's a popular way of printing output. So here it's just saying, okay, now print column 1, $1, then a space between the double quotes, then print column 2, and then slash n, which means a new line. And that's it. And that's how we convert the entire file from one format to the other. Does that make sense? So, so maybe you can now try to download the reformatted file. You can, so if you click on the where there is like 17 here, yeah, no, right there, yeah. You can see there is a little icon with a floppy disk. Okay, so you see this is the same file. So if you connect now to
I didn't do this step, so if someone else already did it.
It's super fast. So just go ahead.
Okay. Okay.
So just, now just drag and drop the
Find it.
file wherever it is. You need to, yeah, it's in your download maybe. There you go. Okay, now you can change, you see that you can decide which K-Mer length. In this case, we use 21, I believe, so you just keep 21. You can have the deployed that in this case, it's equalized, so it's going to be 1. And you have a bunch of other options here that are relevant. So the max K-Mer coverage, of course, it's basically allowing you to filter out very high frequency stuff. If you imagine to have a contamination from some, let's say you have a vertebrate genome and you have contamination from bacteria, you may want to get rid of the ultra high frequency stuff that may affect your analysis. But in this case, you can do it as it is. And then there is this average K-Mer coverage for a polyploid genome, because as we said, estimating these parameters for polyploid genomes is very complicated. So here you can actually provide a estimate that you already have, because you know approximately the coverage of reads that you have generated for the genome. But again, in this case, it's gonna work fine. I do it. So submit.
This is fine as is, or I'm sorry, did you say change it to 1 or it's fine?
No, -1 means do not try to estimate or do not fill.
Okay.
So submit. Continue. And again, it's really fast, but you can see it was actually too fast, because otherwise you could have seen that it was trying to fit the model. So here we have only one peak, right? Because instead of being a diploid organism, it's actually a haploid organism. So one peak, the GenomeScope made a decent job in trying to fit the peak. You can see that it's not really completely covered. Now there is like a blue stuff on the top, but the reason for that is that we're fitting a model. So in the real world situation, the data does not follow exactly the expectation, and you may end up with results like this. Also, you can see here is a very high coverage, of course, because when we sequence, or whoever sequenced this data set, which is E. coli, even with a relatively low throughput machine, you get a ton of coverage for it. So here it's almost 300x coverage in terms of K-Mer, which is approximately also the same coverage in terms of sequencing depth. Okay. You can see, of course, that the frequency of K-Mers is much lower, because again, we're talking about something that is small. So then the total number of times you can actually, the total number of different K-Mers that you can actually see in that particular genome is not particularly high. We have like several order of magnitudes lower than the other case. And you can also see that there's a clear separation between the error stuff and the actual genome. Okay, this is very good, because we can then use this information in later stages to really understand exactly what is an error in the assembly, what is actually good in the assembly. And we're going to talk about this afterwards. So then, of course, if you scroll down a bit, you have the same plot with the high frequency stuff. Okay, and here again, a bit confusing. I would say the take-home message here is that there is not too much highly repetitive stuff. And you can see, in fact, that the unique stuff is 99.3%, yeah, as predicted here. So, And of course, it's haploid, so 100% haploid. Okay, that's cool. Anyone by chance tried the yeast or the Drosophila? No. Well, too bad. So thank you, Sadie. I'm gonna share my screen again. By the way, mine is still running the assembly, but I have computed the K-Mers from the reads and the FastQC. So how many people are familiar with the FastQC output? You know what FastQC is? Because it's this one. No, never run a FastQC on an NGS dataset of yours?
I did, but like 10 years ago.
Okay, yeah, that's true, because actually FastQC, it's a software that has been out there for a while. They have this versioning number. I don't know if you see it here down the bottom. It's still 0.11, but it's been there for a long time now. Okay, so the way FastQC works is that it's a combination of a tool, basically of algorithms that are going to evaluate different properties of your row reads. Okay, so you take your row read data set. It's actually being developed mostly for NGS data, so for short reads, Illumina. But it actually works, as you can see now here, it actually works almost identically if you are using long reads, okay? So you get these report here about the total number of sequences. So we have, I don't know if it's big enough for me. Right. So total number of sequences, literally means total number of reads. And of course, here I'm using the same E. coli as Sadie was using. So it's not much, because I mean, with 100,000 reads, a 5 million base pair genome can actually be, with of course, long reads can actually be covered pretty well. And you can see that the sequence length goes between 1,000 and 20,000. Okay, so that's a typical run for hi-fi. Well, I mean, I don't know, Olivier, is that a typical run?
Yeah, I don't know. That seems shy, but
yeah, but also here it's a relatively old data set, so they're not aggressively size selecting.
it's 1 KB to 20 KB. Yeah.
The small stuff is still in there, it seems, but here, of course, it's a range, so we have to look at the distribution.
Yeah, to be honest, I never run FastQC on hi-fi data. But
yeah, well, anyway, so the GC content is 50%. This is amazing.
oh, one thing I would like to point out,
Yeah.
you guys seen that with short reads, it looks like? With short reads, you will see the curve going down, meaning the quality drops down along the reads, right? You have a bias to the end with low quality. You don't see that with PacBio. Right? You see a pretty flat line.
That's a very good point. I always found that this plot a bit misleading, because what they did is that they show you the first 10 bases, then you have a range of 500 bases, then you have a like big compressed ranges, so you get the idea that, and this is made, I think, because it doesn't matter too much if you're looking into short reads. They are still doing that, so you have the first 10 bases, then you have I don't know, 20, 50, and so forth. But here, I think it's really confusing, because then you have this impression that this counts as the rest, but it doesn't. So, but anyway, here you have the quality score. No? It's based on the position in base pair on the read, and of course the first 10 are not going to be a good average as the rest, but you can see that hi-fi reads are pretty stable and in the I range, no? Because the green stuff here is also where the good Illumina run is. So actually, we're talking about reads that are 20 KB long, comparable in quality to the Illumina or Sanger, as it says here. Okay, so that's good. Then, of course, here there is something that is specific of hi-fi reads. We're talking about very long reads, and as you can see, the mean sequence quality is not necessarily always that high. And you have a very high peak where the mean quality is actually high, but you tend to see a I would call it a smear of reads where the mean sequence quality is not as good. It depends. I mean, I think the quality today of a hi-fi, this is not necessarily very recent data set. I think it's now getting better. So you have a very, you have actually apply rules to filter out this stuff, basically. And so you get a much better and much tighter distribution here. But again, it's more a mixed population of reads. And this is because if you remember how the hi-fi reads are generated by this consensus approach, you go in circles around the molecule. It's again, it's a stochastic process, so it depends how many times you actually sequence the same molecule. Especially, it could also depend on the length of the molecule. So if by chance your fragment is 10 KB, you may go around it 5 times. So if it's 20 KB, you only go around it 2 times. So of course, PacBio and the library prep step have been optimized to try to reduce this variability, but there is still some in the prep. This doesn't mean that these sequences are bad. I mean, these are also good, but of course, the quality is not as good as in the other case. Well, the per base sequence content is not necessarily very interesting most of the time. It's usually in Illumina reads, it's actually interesting to see if there is something at the beginning of the reads that may be due to barcodes, restriction enzyme, or similar. But you can see that as expected, all bases, P, C, A, G, when you walk through the read, they're all present at about the same proportion, especially when you start to average, as in this case, between 500.
I find interesting that at the end you lose some randomness a little bit at each extremity. That's interesting.
Right. Well, I suspect here there is something about the adapter or similar that it's
maybe it's something completely clipped. Yeah.
Well, this is also something that I've never found really informative. I mean, if your real peak, GC peak, the red one, goes somewhat, I mean, it's overlaps somewhat with the theoretical distribution. That seems, let's say, reassuring, but I wouldn't draw many conclusions from this plot. There are no ends. That's a canonical file from PacBio. So Illumina can have ends. Most of the reads with the ends are actually removed by the machine or in downstream processing, but you can still have ends. And in PacBio, no, that's not expected. Sequence length distribution, again, you have a somewhat weird distribution. So you have a range. You remember that it was telling us that there are some few reads that are very short. As you can see, the vast majority of the reads is not shorter than 11 something, 12 kilobases. Okay, you have a very strong peak at 14 kilobases, and you have a tail of some stuff that gets almost up to 20 kilobases. You can play depending on how you prepare the library on this, so you can have you can shift the distribution. You can get longer reads, shorter ones. It depends. This is being used not just for Genome Assembly, but also to sequence amplicons. So if you have I don't know, a region of interest that is, let's say, 2 kilobases, you can maybe generate amplicons of 2 KB and then only sequence those with PacBio. And of course, your peak would be around here. Well, the sequence duplications levels, I mean, are of course close to nothing in PacBio, because I mean this is something that is typical in Illumina reads, where you tend to have many duplicated sequences. This is important because when you sequence with Illumina, you have the bias of PCR that may actually generate more copies of some stuff, and so you want to check that. But here, of course, we are talking about very long reads, and they are almost all different. Then there is nothing that is overrepresented, so it means that it seems that we don't have any major contaminant. But again, we're talking about E. coli here, so normally E. coli would be the contaminant of some higher organism, of course.
Yeah, but after we're talking about like 15 KB reads, so the chance of finding the same reads multiple times.
Well, this would be for the sequence duplication.
Yeah.
Over,
Oh yeah.
It's,
right.
it's just if there is a barcode or some contaminant.
Yeah.
So it doesn't matter if it's
Or repeats or stuff like that. Yeah.
But again, here there is nothing. Adapter, no adapter, but here it doesn't count because these adapters that are checked by default by FastQC are only Illumina adapters. So you would have to add to the library the PacBio specific adapters to actually understand what's going on. Okay, so that's the report. You can see that here is telling you where you failed or where you didn't fail. But again, it depends on what you're looking at. So sometimes it's suggesting that there is a problem, but there is no problem. Here I don't see any real problem. I don't even know exactly how it's predicted that it should be. Maybe the distribution should be like that, but yeah, again, I wouldn't worry about it. And of course, again, here it's tailored on Illumina stuff, so this is not really something you should worry about. Okay, we have maybe.
So about the adapter sequences, they are still in there now, right?
Well, so normally, if you had contamination from adapters in Illumina, this would be like spikes at the beginning or at the end of the position reads in the base pair. In this case, so with hi-fi reads, I don't know if this particular data set contains some leftover of adapters of PacBio.
You will see them in the middle, right? Because you will see the adapters.
Normally, if there is little contamination, you really don't see them. But if it's like high level of contamination, it would pop up as an overrepresented sequences. So yes, it's unlikely that there is a significant contamination at this point.
In theory, they're all clipped already, but yeah.
Yeah, but the best way to do is actually to update the internal FastQC library so that it also checks for adapters from hi-fi reads, for hi-fi reads, and so you can actually see if they occur in this block or not.
So at this step, they were clipped already, the adapter sequences?
Yeah, so the PacBio should in principle remove them when let's say when the output is generated, so they provide you adapter-free sequences. It's not always the case, and it turns out that
there was about 1% left, or very little.
Okay.
Sometimes there is this 1%, and actually it's a problem. You want to check for this because it actually increases the chances you make mistakes during the assembly process. So you want to, in principle, always check and get rid of them if you can.
So most of the time, these adapters are going to cluster together and create this kind of weird little config. But we've seen some cases where they got incorporated into the real contigs. So you have to make sure.
Actually, yeah, when you submit an assembly to a CBI, for instance, they check for adapter contamination because it's very common. And again, it could be separate sequence so they can get rid of easily, or it could be that somehow it made it into the sequence of chromosome 1, and so they masked it. So they put ends in that position. Yeah, there is a lot of junk in the assemblies that are submitted to the archives. So yeah, they have to pay a lot of attention to try to avoid I mean, junk, I'm not saying that people are submitting junk. I'm saying that within a submission, there's maybe a good assembly, but there are a lot of small contaminations. Maybe you get, you know, and you don't know that there is a symbiont that is a part of the sample that you have sequence and assemble, and you have you know, organelles from the symbiont, the entire genome of the symbiont submitted at the same time. So they also have these contamination screenings to try to see if they're actually submitting multiple species at a time rather than a single species. Yeah. Okay, so these, I think it's pretty much exhausted about the FastQC. I think we can take a break for 10 minutes, and then we have a couple more things to say that I think are useful here. And yeah, so see you in 10 minutes.
Sounds good.
3:30.
No pressure. Okay, is everybody there? Wanna turn your camera on when you're there, so that we don't feel too lonely? Well, Jose, I'm sorry, you said that you were able to run the workflow, right? Also Busco? So maybe we can look at your results afterwards. Okay. Jean, Nicolas, Sadie, are you back? Is everybody back?
Yes.
Okay, thanks. Sadie, you're swimming in your pool?
Yeah, she's finishing a pina colada and she's coming.
Yeah. Should have I known that, I should have also gone to Florida for the pandemic. They said it wasn't a good idea, but actually it doesn't look like that. Well, okay, let's this is pretty simple, so I'm gonna get started again. So well, this question, I hope we had already kind of answered by now, also with the theoretical part of the course. But just to give you a proper definition, or a definition I like, genome assembly is the entire genomic sequence derived through a de novo, which means reference-free, because there are people and there are approaches that say to reconstruct assemblies or something that looks like an assembly, but rather realigning the reads to a reference and then changing the reference based on the read pileup. So that would be a reference-based assembly if you want, or reference guided assembly. But I would say that the real genome assembly is when it's de novo. So when you actually have all the row reads, all the pieces, and try to reconstruct the puzzle without any additional information. So it's an assembly process of the row sequencing reads, and then it gets released by the curators of the genome in a database. So we're talking about something that is published in the, let's say, GenBank or in other databases available in the world. And for now, we have almost always been looking at reference genome assemblies that correspond to a single individual or even pooled individuals. Now, the first human genome was actually derived from multiple individuals. And of course, this introduces complications. But normally we're talking about, at this point, one individual. And its haploid always haploid, despite the fact that in most cases the species is not haploid, but actually we want to represent only one of the two haplotypes. The reason for that is mostly, I would say, historical, but also practical in terms of, well, historical, because until recently, it was not possible to assemble diploid genomes as diploid assemblies. So you basically would have collapsed the two copies into a single copy, and then you would have reported whatever variant present, let's say, in the paternal haplotypes versus the maternal haplotype as a variant, okay, whenever you were able to identify those. The other reason is practical in that for many applications, haploid assembly is more useful, because you have, let's say you want to map your resequencing reads from 100 individuals to understand the variability in a population. You don't need two copies of the reference. Actually, that would be more, it would actually introduce more complications than solving issues, because you will have two exact copy or almost exact copy of the same thing, and then you would not know exactly where the read should go to, if it will copy one or copy two. In fact, the diploid genomes are often described as being a double, like a gigantic repeat. Now you have a repeat copy of the same assembly twice, or even more than twice if we're talking about tetraploids or beyond. So again, most reference almost all reference are haploid. We're now leaning toward this idea of pan-reference genomes. So the idea that there is some variation that is typical, there's a relatively large variation is typical of some individuals but absent into other individuals. That's very common in metagenomics. So when you're dealing with bacteria that recombinate, that can exchange genes and so forth. So instead of, or even like in rice, where there are some regions of the genome that are really absent in some individuals and present in other individuals. So there is this idea of creating pan-reference genomes. In plants, and some plants are actually pretty advanced, or in metagenomics, but now there is also the idea to apply this to human beings with, for instance, a project Olivier and I and others are involved, the Panhuman reference genome, to create a reference that is actually more representative of the whole diversity of human beings. As you know, there is in the current reference a lot of bias, and it's not representative of, I don't know, Korean variability, for instance. So yeah, and at this point at present, we report these variants as alternative alleles in what is called a VCF file. But the idea is that we can actually probably have a more complicated but more informative, essentially, format that can contain all this variability in a single reference. And therefore, when you map your reads to this reference, you can actually imagine that you identify everything, because you have all the let's say, even the what it's called the accessory genome, so the stuff that is maybe present in some individuals but not all individuals. The other problem is that these haploid assemblies, especially again the previous generation, but it's still quite common, they are completely shuffled. So the haplotypes are completely mixed. So you're not really even representing one single haplotype. You're not representing chromosome 1 from the mother or chromosome 1 from the father. You're actually representing a chimeric chromosome that is a combination of the alleles that were present in the mother and in the father at the same time. And they're, just walk through the sequence of the chromosome, and they are occurring almost randomly. So the idea of phasing the genomes, it's about trying to separate this information and really reconstruct a chromosome 1, and let's say, or chromosome 2, or whatever, that actually is representative of the real sequence of the chromosome of the mother or the father. As of now, that's really limited. There are a handful of genomes that actually have done that. Okay.
Join you?
Sure, go ahead.
Did you say that common variants are usually reported in reference genomes?
Reported as variant files. So it means that you have I mean, it really depends. I mean, there are multiple ways. But for instance, if you take the Human Genome Project, so they assembled one reference, and then the 1,000 genome projects through resequencing assessed not just 1,000, actually 2,000 and something individuals. Okay, but then they don't reconstruct all the sequences for all the individuals. What they only tell you is that, okay, this individual with respect to the reference had a G here, a T there, an indel here, and so forth.
So for that, you need to have genomes of several individuals. So for the org, right?
Well, actually, I mean, at least one individual. If you want to have at least some variation with respect to the reference, you need one individual that differs from the reference, and you can list the variants of that individual.
Yeah. So if you have organisms that were not sequenced multiple times, like I don't know, a non-model organism that was sequenced only once, there will be no information on any possible variant, right?
Well, you are still in the position to identify the variants from the alternative haplotype. Okay, so you represent, let's say, the maternal alleles. Okay, you know where there is a difference because you see that the reads are 50-50. You know, they are 50% A, the mother, and 50% T. So in the reference, you put the A for the mother because you decided so. And then you know that there is a T, so there is a separate file where say at that position there was a T, and that it's likely the paternal contribution.
I see. So you will normally have two possible genomes, even if only one was sequenced for a diploid organism.
Yes, yes. And depending on the approach you use, you can really have two. I mean, with some sophisticated approach, but again, is a handful of genomes, you can really separate them so well that you actually end up with really two genomes, one from the mother, one from the father. We have some slides on this.
Good, thanks.
You're welcome. So I'm going to walk through a bunch of slides. Maybe we can then check the boost code. No, there is a slide on boost code, so maybe we can get there first. So, and here we come to basically your question here. So normally when you go to CBI, they define something called the primary assembly. Okay, the primary assembly is in GenBank. So on CBI, it's what the curators, whoever generated the genome and CBI consider the up-to-date resource for that particular species. Okay, and this is also the first result that you see for this primary assembly. I'm sure you may be familiar with this acronym here, Genome Reference Consortium Union Build number 38. So this is the you know, the descendant of the original genome for the human reference. Okay, we are I think they skip numbers, so it's not really accurate to say that it's the 30th assembly, but we're of course, since 2001, there has been a lot of different releases over time. I think actually probably this is the, I think it's from 2013. Anyway, there is the sequence here. So if you get into the website and navigate at some point, you may see something like this, where it's in this case, for a particular gene, it's going to tell you the coordinates along the reference where you can actually find that particular gene, and you can download the FASTA sequence for it. Okay. Of course, in order to know where the coordinates of a gene are, you need an annotation. This is a whole, a completely different world, let's say, with respect to what we've been discussing as of up to this point. Because we're just been talking about assembling the genome, but okay, well, when you assemble the genome, the next step is, okay, let's try to identify features of this genome. So that's the annotation process, where you find, first of all, protein coding sequence, but then you can have, long coding RNA, small RNAs. You can have repeats that are relevant. You can have transposable elements, all this kind of stuff, and it's a whole separate world, if you want, with respect to the annotation process. So when you look into the record for the primary assembly for GRC H38, you can see, well, it was, 2017, sorry. And you can see that it says haploid without loci. Okay, so this is a pretty accurate one. It's a haploid, but also they report the variation for the other parent, basically. Okay, it's chromosomal level, so they assemble or reconstructed all the chromosomes. Of course, it's a reference genome. It's complete, full representation. You have the accession numbers that are important to, of course, download or even refer or just reference this particular assembly. And then you get some other information. You see also that they tell you that there are a lot of other assemblies for human. Actually, GRC H38, it's pretty good, but the one that we presented last time for the telomere to telomere consortium is actually better than this one. The problem is that whenever you change the reference, everything has to change accordingly. For instance, the annotation changes, so there is a lot of it's a small revolution. So people tend to stick to older and less accurate, in most cases, versions of the assembly, because maybe they have established pipelines and they know the approximate coordinates. And if the coordinate system changes a bit, everything gets confused. So they tend to stick to something that is maybe less accurate. But anyway, this is a pretty good assembly, and this is the standard reference. You can see measurements of quality, high-level measurements of quality, let's say, or these metrics related to the assembly. So here, for instance, you can see that it says, well, the total sequence length is 3 gig and almost and 100 meg. And this is pretty much accurate. Again, it's not complete because no genome assembly, even for human beings, let's say for a higher organism, no genome assembly is complete. For bacteria it is, but that's a different story. But this is pretty complete, pretty close to the actual sequence. There are some gaps because you can see that the total gap in length is actually much smaller. This is because especially the very nasty repeats at the centromeric level, they know where they are, they know approximately how long they are, but actually they were not assembled, so that's why they are large, relatively large gaps. We're talking about 150 megabases. And when we described the telomere to telomere assembly, we said that there was recovered about 200 megs of that sequence. So it means basically all these gaps were filled. That's why I'm saying that assembly is better than this one at this point. Olivier, you have a...
Oh, you said everything.
I'm talking too much. Okay. Well, so gaps between scaffolds. So there are 350 gaps, 349 gaps overall, which again means, I don't know, some 10, 20 gaps per chromosome. Number of scaffolds, 472. Well, that's a bit misleading because here we are also including, I think, the some alternate stuff. And then we have these magic number, the N50. The N50 applies, it's a metric that I think was developed in some hackathon. It's almost like the median value for, in statistics, but it's not, and it's confusing, and it's very popular in bioinformatics, but it's actually really weird. I mean, in terms of the behavior it has. However, you better know what it is because it's everywhere. And it can be simply defined as the sequence length of the shortest contig at 50% of the total genome length, which I think it's almost impossible to understand like this, but with a picture is actually quite easy. So the idea is that it measures the contiguity of the assembly. So how large the chunks of the assembly, let's say, the chromosomes are. And you could apply it to anything. You could apply it to the row reads to measure approximately how large the reads are, how long the reads are, or you could apply it to the chromosomes, or you could apply it to intermediate steps of the assembly to evaluate how contiguous your assembly looks like. The idea is that you have, in this case, let's say a fictional example with 7 pieces. The first piece is 100 megabases. The second one is 70 megabases, 60, 50, 50, 40, and 30. Let's say these are the 7 chromosomes of an unknown species, okay? We order them from the largest, sorry, to the smallest, and then we sum their lengths, okay? So the total length is 400 megabases, which is pretty much the size of the example with genome scope, but it's just by chance. And then we compute alpha of that. So alpha of 400 is 200. And then we say, okay, so if we start to sum up the length of starting from the longest to the shortest, and we stop when we reach half of the total length, okay, so 100 plus 70 plus 60, we actually, when we stop it, because if we we consider the first one is 100. We sum 70, 170, we're not there yet. So we need to also add 60. When we add 60, we are beyond 200, and that's where we stop. And we consider this particular value the so-called N50. So in this case, the N50 is 60 megabases. I think you can imagine how confusing this is if you then have to compare these numbers between assemblies. But there is, I mean, this is used everywhere by everyone, and so it's something that, I mean, you must know. For some application, it makes sense, but it's really, I mean, it can really be defined.
So one thing you can imagine, this estimate is going to plateau after a while, right? Because chromosomes have a maximum size. So depending on your genome size and your number of chromosomes, these values are going to plateau.
Yes, it expresses a lot of weird behaviors in different situations. That's why it's kind of tricky to use. It's being used because basically it ignores all this small stuff. So imagine that instead of the last piece here being 30 megabases, imagine that this was broken down into 1 million pieces, okay? So each piece of 30 bases, okay? 1 million pieces, 30 bases, each is 30 million. The N50 wouldn't change because everything that is small doesn't count. Okay, so that's why it's been used. It's popular. You ignore that, but I mean, with that, I mean, it comes at the price of a lot of potential weird outcomes. But anyway, as you can see, the main information, one of the let's say 10 most basic information from the assembly reported by NCBI is actually this N50. And in this case, it's 70 megabases. So it means that about, let's say, half of the assembly is in pieces that are bigger than 67 megabases.
That's pretty close to chromosome length. I mean, it's very close. Yeah.
And yeah, but there are yeah. So because of course in here they're constructed the entire chromosome, so you would expect that to be the case.
So that would be considered perfect, right? If you reach the size of the chromosome,
Correct. Yep.
It cannot be more than that.
Yeah.
Yeah.
The problem is that you may reach the size of the chromosome and have the sequence wrong anyway. But yeah, that's the idea. You have two different measurements here. You have the scaffold N50 and the contig N50. If you remember that the difference between the two that was explained by Olivier last time is that the contig is where there is no gap. No, it's the sequence. You find the overlaps between the reads, and then you reconstruct the sequence resulting from these overlaps. But then, even if you have, let's say, repeats of the boundaries of these overlaps, so you don't know where to go, you have other approaches like the bio-nano optical mapping, or HiC, that allows you to basically put these pieces together, but with gaps between them, and that's how you get to scaffolds. So of course, the contingent 50 is going to be smaller than the scaffold and 50 because these are the smaller chunks that constitute actually the scaffolds. And then you have a lot of in this case, you have one plasmid with the mitochondrial DNA, okay, 24, 23 plus 1. But of course, in a bacteria, you could have a ton of other sequence. Okay. And here we come to Busco. I mean, this is just about the Phred score. I don't know if that's something relevant here. Could be. Do you know what the Phred score is? You want to tell us? No. Okay. Well, okay. So the Phred score, I'm gonna say it quickly. It's something that was introduced with Sanger sequencing. At some point, people were realizing that they were making a lot of mistakes in calling the bases from Sanger sequencing when they were doing it by hand. So they said, okay, let's try to introduce something to automate this process so that we call the bases, but also when we call it using an algorithm in an automated fashion, we actually associate an error to that particular call. And so the idea here is that you can actually get approximately a probability of that particular base being wrong, okay, and that's P. So let's say, and then the Phred score is just the logger of that probability to the -10, so times -10, and so it means that you transform it into a log scale where if you have 10, you have 1 error every 10 bases on average, so 90% accuracy. If you have 20, it's 1 error every 100 bases, 99% accuracy, 30, and so forth. Currently, the standard is that you should, with the quality of your assembly, means how many errors per base there are on average, you should at least reach this QD40. It's considered kind of a theoretical boundary because it should guarantee that you don't have many or very few errors for each exon in your coding sequence. And if you have very few errors in your coding sequence, you will not have frame shifts that mimic, stop codons or something like that. So when you annotate the genes, your annotation is of good quality. So that's the reason why. But it's also not really easy to measure this number accurately. So it's still it's there, but it's the point is that we are actually applying this principle developed for Sanger sequencing also to evaluate the consensus accuracy of the assembly itself. Anyway, let's go back to Busco. I think Olivier, this is for you.
Oh, so thank you, Julia. So Busco essentially helps you to understand the completeness of your genome. And the way it does is going to look for some genes that Busco has a database for, that gene that we know that are unique copies and they're present in every genomes for certain clade. So Busco is looking for these genes using BLAST essentially. And what it does is going to output some stats like completeness. So how many genes did he find, right? So in the first row here, you can see there were 750 genes he was able to identify in your genome that he was expected to see. 715 has single copy, but he sees a lot of duplication. 35 of them are duplicated. So this gives you an idea about either these duplications are real, is biological, or probably they're due to like some assembly errors that where different parts of your genome is including twice. F means fragmented, so it tells you if you have only a piece of the gene. How many are missing from the total number of genes that here in this particular case was 1,000. So all these stats give you an idea of how complete things are and how wrong they could be. So it's a combination of both.
Yep.
Any questions about Busco scores? So Busco score, you use different database depending on the species you're using. Now, I mean, I don't always believe Busco score. They tend to also to plateau when all the genomes are good. You don't see much difference between a good assembly and a bad assembly. Now things are, it's less and less useful to me. But I don't know you, Giulio, but.
No, it's definitely so. The idea is that you generate these assemblies that are de novo. So in most cases you know nothing about them. No? You don't know exactly how to describe. So Busco was developed as a way to quickly assess the quality by saying, okay, if I have 1,000 genes that I'm sure or almost sure that they should be present in my sample because they are highly conserved, so they are found in almost all species of that group, that taxa. Then I'm expecting them to be present in my assembly too. If they are not present, well, there is maybe something wrong with the analysis, or my assembly is really bad, or depending on how many you find, actually could be more or less bad. So yeah, so the problem is that it's not true that all the genes, even those that are highly conserved, are necessarily present in all species.
Or highly conserved in the species you're doing, right?
Yeah.
Yeah.
So that's one problem. Then there are other problems in terms
or gene duplication, genome duplication, polyploidy, all this stuff will influence your Busco score.
Yeah.
You have to take it very carefully, but.
So normally, anyway, I mean, it's an indication of something going on, right? So if you have a lot of missing genes, you should be worried, most likely. If you have let's say 10%, you're still you can still have hope. But if you have 99%, I mean, that's good. It doesn't mean that you're not missing something, but it's good.
Most of the time, the duplication is what you want to look at. The D, meaning that you have an assembly error somewhere. And Judo can talk about that at the next step after the assembly, right?
Yeah, because here the idea is that the genes that are used by Busco, that are being selected for Busco analysis, are expected to be, again, it's not necessarily always true, but they're expected to be single copy genes. So genes that are not found more than once in any species. Again, not necessarily always true, but most of the time it is. And therefore, if you see a lot of duplicated genes, it means essentially that your assembly is not haploid, that you have represented 2 copies, for instance, or even more of the same gene, of the same region, of the same sequence in your assembly. And these, as we said, it's not actually a good thing, because when you want to, I don't know, map reads, if you have 2 copies of the same gene and it's meant to be haploid, it's basically deployed for that particular gene. So when you try to map your reads, you don't know whether they should go to one copy or the other copy. So that confounds the alignment, for instance. But there are other reasons for that. So this is something to check for. And there are tools that are very important in assembly pipelines to get rid of these duplications. The reason why these duplications are introduced in the first place, it's a whole chapter of its own, but the idea is that basically the reads contain errors, and we know approximately what the error rate is. But sometimes the error rate is just by chance a little bit higher, so you get some reads that belong to the same gene, okay? They correspond to the same gene, but compared to a certain set of reads, they have a higher error rate, and so they do not cluster basically together with those reads. And the assembler says, okay, I mean, they're not good enough, they're not similar enough to contribute to the same thing. So I'm going to generate a new contig that corresponds to these reads that are different enough to do so.
Yeah, another possibility is if you have very heterozygous genome and your gene is very deployed, very different. It may be included in the primary assembly instead of putting aside as alternate haplotype. So what you're going to get, you get the copy of the 2 same thing. it's a routine haplotype in the primary.
Exactly. So actually, this is probably the more common reason, I would say. It's the same principle, but here the idea is that it's just so different that the alternate haplotype is just so different. It gets separated, but you don't realize it's a different haplotype, and you include it into the same.
And Busco thing is the same thing. So there is a different sensitivity between Busco and the assembler too. Yeah. So.
Yeah. So here you see
Busco performing on bird genomes, for example.
how is it performing?
Yeah, with bird genomes.
You see some of these duplications.
Yeah.
So for all our assemblies, we have to do what we call purging. So we can identify this duplication. I mean, Julia, are we going to talk about this?
There is a slide. Yeah.
Yeah. So we can talk about it later, how we remove them. But yeah, we see that in birds, yeah. Especially in wild caught birds. I mean, I don't think we see that much in zebrafinch, but maybe I'm wrong. But
No, I mean, birds are good. Birds are easy, considered the easiest vertebrates on average to assemble. Small genome, not a lot of repeats, relatively simple to assemble. Yeah.
yeah, you can assemble a bird genome within 3 hours.
Yeah. So
That's great news.
But if you want to assemble a lot of them, let us know. So here you can also see that there are different specimens. You have different numbers of Busco genes because you are actually using different Busco gene sets depending on the taxon. And you, of course, get different values of Busco and the different number of Busco genes recovered. Now, this is a pretty bad one. Of course, I don't know what it is, but it's pretty bad. This is the best one here. I fear we've lost our chance to look into a real Busco result, unless Sadie, you have run it.
Sure, I can show you what I have. I would love to hear how you interpret it.
Absolutely.
Oh wait, you have to let me share the screen. I think it got disabled.
Matthew, us.
Okay, there. Okay, let's move you to the side. Okay. So let's see. So this is the primary. Let me do this. Okay.
So this is the full table because Galaxy is providing you also with the full table of the results. So as you can see here, you have all the genes basically that were found now, and you have also the description of them on the right. And you have an ID from this ortho database, which basically try to maps even between species the same genes using a common nomenclature. And you can hear it's telling you on which sequence you actually find it. You see this PTG001 there, it's actually where. And it's also telling you the coordinates where the gene was found on that particular sequence. And you can see it says also the status that it's complete. You should have also a summary, a report, because this is, again, the full table, but it's not very, and the summary should be another output from full table. No, do you have, it's a, should be a completely, no, it's, yeah, so this is Galaxy, so you can actually try to visualize your data within Galaxy running some of their pipelines, but no, I'm saying that there should be another result with Busco, but this is a full, no, it should says summary, maybe scrolling down.
Keep going.
Yeah, keep going.
Summaries. No.
Oh, it's
It's a different thing.
Maybe, I mean, maybe I missed if there was a step.
No, it should be.
Normally it should be with these, you have something deleted. Maybe you deleted it by mistake.
I don't know. I mean, I know I looked at the alternate, which in this case with E. coli, there's nothing, right? Because we already saw two, or maybe there's one thing.
Oh, no, I know, but yeah, can you click on, maybe you deleted it or hidden, there's something hidden on
What do
top.
you click on? I'm sorry, I missed it.
No, if you, under a named history on top, there is 16 shown, 17 deleted, 2 hidden. Now, you don't have to scroll, it's right there where there is the gray area. No, it's up,
Oh, okay.
up in the page. Up in the page. Here, a named history, 16, 17 deleted.
Well, these are the ones that I think, how do I look at these?
I think you have to specify in advanced options that you want the summary. I
Oh,
think.
yeah, it's true. I forgot to, because
That might-
what happened is that they upgraded from Busco 4 to Busco 5. And I told them, please leave as default that there is a summary table, but the guy didn't listen, and they actually put it not as default option. So also my Busco results are not going to contain it. So
Open Busco, and if you click on advanced options, and you go down, and you will see which outputs should be generated. You click on select, and now you have short summary text.
yeah, that doesn't make sense. I told them, but they didn't listen.
That's what it is. I could do it on the primary, for example, and then, but I don't know how long it'll take to run it.
It should
Okay.
be fast. It should be fast.
Yeah, that makes sense. 'Cause I was like, I looked at it, I was like, that's why I was like, I really want to know what you think, how to interpret this, because it's full of data. I'm like, it's interesting. I don't know, how do I quantify? I mean, it says complete for all of them, so I'm assuming that they're all there, but.
I mean, I'm sure you can take the output in Galaxy and calculate the frequency for each column. But
Do you have it, Julie, already done? If you want to show yours.
Actually, I don't.
I'll see if it runs, but maybe it might take a little bit.
I think it's gonna make it. It's very fast actually at this point. Have you selected the right species when running it?
I think you use the defaults, Eddie. Did you?
Let me check.
Make sure that you're using the actino, the protobacteria.
No, you're right. I didn't. Good catch. I did not change it. It didn't keep it. Okay.
So
Now we get to see all over again.
Meantime, mine is still running the assembly. Of course, the time it takes to run, it really depends on resources. So here we're using 3 resources from Germany, but if you have your own resources, it can be much faster. It depends on how many CPUs you basically allocate to the job. But I5 reads are actually assembling very, very fast. So let's walk quickly through the other few slides. You wanna take it? I mean, you want to present this, Olivier?
Sure. So this is the IC plots. So it's essentially what you do. I don't know if you remember IC. IC is the cross-linking of DNA, and then you sequence the junctions of the cross-link, right? So you expect to see most of, and after you map it back to the genome, and you expect to see most of the fragment to map on the same, on contiguous sequences. So once you have your genome assembled, you can do the same thing. You map your IC data to visualize all the junctions. And here you observe something you expect, that where the diagonal with red, yellow, and darker blue has a lot of mapping in these areas, right? Because diagonal is where all the sequence is contiguous, right?
Yeah, so here,
yeah, go ahead.
Because here you're comparing the assembly against itself.
Yeah, right.
So on the X and Y axis, you have the same scaffolds, okay, the chromosomes. So chromosome 1, let's say chromosome 1, column 1, chromosome 2, chromosome 3, and so forth, and the same on the other, on the rows, okay? So basically, this box here corresponds to chromosome 1 against itself.
Right.
And you would expect to see that the mapping is within the same chromosome if you actually reconstructed the chromosome correctly. And you shouldn't see reads that map between chromosomes if the chromosomes again are correctly reconstructed. So here, for instance, you can see a problem where there is a small chunk here that it contains a signal connecting the chunk, this small chunk here, to chromosome 4, let's say. Okay, so this is a problem where basically this chunk here, which is probably a telomeric region, a subtelomeric region of the chromosome, was not assembled correctly, and it was left basically alone and was not integrated into the chromosome. But of course, now that you know that, you can actually place it manually or using software that can take advantage of this kind of information to try to basically put it back to where it belongs.
This is what we do at the end of the assemblies. There's a collaborator that do what they call curation, where you look at this kind of information and move the pieces around to reconcile the signal.
But this is pretty amazing already. I think this is a bird, and it's.
Oh, you should see Greg's budgie.
I showed me that. Yeah, it's perfect. So the thing here is that this is the results of the automated pipeline, and basically we have reconstructed almost all the chromosomes, with the exception of a few small chunks that maybe belong to the chromosomes that were not assigned to them, just from the automated pipeline. So that's.
So of course you see on the lower end, you see a bunch of, is very green. So a lot of them map to each other. It's probably because these contigs that were not incorporated, or they're macrochromosomes, or stuff very repetitive. Usually what we do, we know the karyotype, so we're counting number of chromosomes, and below that, okay, that's just mess. So we're counting on any chromosome.
Yeah. So the thing is that then if you have a karyotype and good images, you can potentially correlate the size in the karyotype with the size of the assembly. And if it matches, you may assign all the chromosomes to their corresponding numbers based on the karyotype. Okay, this is a plot that is similar to the genome scope plot that we discussed extensively. And here, the thing is that basically we took the genome scope plot here, that you don't see the difference between the curves, and you don't have you don't understand that there are multiple things that are piling up on top of each other. They're stacked on top of each other, and therefore they contribute to the final curve. I mean, there is something here about the error that is suggestive, but you don't actually see what's going on. Here, this is a really advanced plot where basically we start to paint these K-Mers that we've seen based on what we see in the assembly. So we see 2 copies of all these K-Mers from the raw reads in the assembly. We assign them to the blue color. We see all these K-Mers here that are 1 copy in the assembly. And by assembly here, we mean a full assembly where we have both the haploid assembly reference, but also the alternate stuff. Because here we're already dealing with assemblies that are quite complete. And so they tend to have both copies, the maternal and paternal copies represented. If that's the case, we expect to see a lot of things that are seen twice in the assembly, and we color, the K-Mers, the corresponding K-Mers, or we color the heterozygous stuff. And of course, you can also color whatever it's not seen in the assembly, and it's also only seen in the reads, and these are the sequencing reads, which is good, because if the black was going well beyond here, it would have sorry, not the black. If let's say there was some stuff that was red, but here it would have implied that there is something that is never seen in the row reads in the assembly, and that would imply that you have introduced some errors in your sequence. And here there is also something that is 3 copy, so it's green, but it's at the same level of the 2 copy peaks. These are the false duplication that we were talking about before. So where you basically duplicate artificially a gene, so it should be present only in 2 copies, one from the mother, one from the father, but you see 3 copies. But actually the support, the coverage support is supporting only 2 of these copies. And this is what you want to get rid of with like a series of tools that take advantage of this kind of, basically because here we have identified them, right? So we know what they are, so we want to get rid of those, but only of those, not of the rest. And you can see that after you run some of these tools, you get a better representation. You see that there is still something, but everything is even more than 4 copies, which was represented here, plus whatever is 3 copies, it's getting is going away. So this means that we have successfully removed these extra copies of genes that we don't want to have in the assembly. Finally, is it done, Sadie? Okay, want to share your screen?
Okay, so I'm running for the alternate, although I don't think it'll be as interesting. So here's the, yeah, so
Yeah.
this, the short summary here.
Which is what we see on the left already.
Okay, Sadie,
Yeah.
what do you think?
So I'm assuming it recovered 100% based on this. So it's saying 366 complete. There was one duplicated one, I guess. And I know you were saying that the duplicated ones are really what we're trying to look for. So I would say that's a really low error rate or a really low percentage of duplicated. So I think this looks great.
Yeah.
And no fragmented?
No fragment.
Nothing is missing? That's a very good assembly.
Okay, so let's see. So it's, oh, single copy, duplicated, fragmented, missing. Got it. Okay, yeah, that looks good.
That's the best assembly ever seen.
It's E. coli. It looks really good.
Well, before the advent of long reads, even E. coli was fragmented and there were missing genes. So this is really the result of long reads that you can have. This is also, if you compute the stats, you will see that this is assembled in a single sequence. So the entire E. coli chromosome is assembled into one sequence. And even if you have a metagenomic sample where you had 20 strains, you would get 20 chromosomes assembled separately with high-five reads at this point.
I mean, we could compare like this one to the alternate. I ran it as well.
Well, the alternate technically is trash here, right? Because the E. coli doesn't have alternate haplotype.
Right. But I did it for the sake of exercise, and I'm saying, I know it's basically junk, but at least we can see here it has all these missing ones. So this would, if this was it, our actual primary, this would be terrible. We recovered nothing.
No, actually, I mean, this shouldn't exist, but as I was saying before, it exists because the data is not perfect. So you will have sometimes something that looks like, oh, here there might be an alternative allele, but even if it's not possible, or it could be because this is millions of E. coli cells that were used. So maybe there are some of them that actually have some variability. It's actually a population, and this variability was somewhat represented as alternate alleles. But again, it's very, very low.
So what you want to do, you want to stitch together the alternate, right? Here the alternate is essentially pieces of the assembly that has an alternate version of it. So what you want to do is to fill the gaps of that, and then you run Busco, right? Because you get a very small fraction. See what I'm saying?
Yeah.
So Yep, that's pretty good. And as you can see, it's also pretty fast.
Oh, it does give you this. Oh, I guess when we pick all of them, oh, that's really big.
Oh yeah.
This is
rough.
where is it? I can't even see it. So where is it? It's somewhere here, I imagine. I don't know. Okay.
Okay. So I mean, probably, as I said, we can finish earlier. I think my run is also completed. And I made it. Let me see my rules stats.
Did you do E. coli as well?
Oh, but I have the full table. Yeah, I think the problem, yeah, it's E. coli, but it's a full table, so it's not really.
Okay.
We run the QV or the Fred score. Do we have that anywhere? That's what we.
No, because it doesn't. So in this pipeline, there is nothing that provides the QV. But we are asked Galaxy to actually implement it, so we're working on it.
So it's just that Galaxy doesn't yet have that as an option.
Yes,
Versus
we
we didn't run it. Okay.
we have to integrate the tool for that. A bunch of other things that I have here that, if you remember, I run is the fast statistics. Again, I have the alternate and the primary. So this is the primary, and you can see that the primary is well, GC content, okay, the N50, and you can see that the N50, the max length, the mean length, the median length, the minimal length, they're all the same number. That's because there is only one chromosome, and it's about the size of E. coli chromosome. So it should be pretty much a complete circularized, no gaps, and so that's pretty good. You see number of sequences 1, and there are no gaps. You see no ends. Okay. You technically can run it also on the alternate. My pipeline was actually doing it, but of course here it doesn't make sense because we're talking about a very the fragmented Sadie was showing, and the Basco score that she was showing. Actually, we're talking about 30 kb overall. No, maybe a bit more. 80 kb. Okay, and this is, let's say, little junk. I mean, it's not necessarily junk. It could be a real sequence in the E. coli pool of cells. You always have this. Now, even if with a zebrafinch, you will have some sequence that is actually maybe mutated in a somatic cell with respect to another somatic cell. So it's possible that you actually get something like that. Okay, so last but not least, this is it looks complicated, but here is basically this is about the haplotype phasing I was talking about before. So the fact that you have, in principle, what you would like to have is a full representation for the maternal chromosome and a full representation for the paternal chromosome. So even with sophisticated tools, you end up not having that, and this is represented here to the right. But with a very specific approach called the trio binding, you can actually achieve that. So you can see, now I'm going to explain what these are, but basically you can see that there is a full separation here on the axis compared to this situation where the bubbles are wandering around the plot. So here the idea is that the bubbles represent the scaffolds or the chromosomes on your assembly. Here are just, it's still not chromosomal levels because you see there are so many bubbles. And then you ask if they belong to the maternal assembly or the paternal assembly, okay, red and blue. And so you color the bubble based on which assembly you have and which assembly they belong to. And then the X and Y axis will place the bubble based on the available information you have from the father and the mother that will tell you whether the sequence within these scaffolds seems to belong to the mother or to the father. It's the similarity basically with the mother and the father. And so essentially here we have only similarity with one haplotype or only similarity with the other haplotype, which means that we fully phase the assembly. In this other plot here, there are some, especially the blue bubbles, that have some sort of intermediate similarity, which means that they were not fully phased. And this is already a pretty advanced way of phasing genomes. But if you, let's say, take a regular assembly from, I don't know, short read sequencing, you will see that your bubbles are really all over the place. So there is no separation at all. And this is the idea. Now, if you have a chromosome and what you want to have is all blue or all red. But here you have blocks where you have a mixed situation. You see short range switches. So you have some errors where basically instead of having only, let's say, paternal sequence, you have something that, according to what you know of the sequence of the mother and the father, belongs to the other haplotype. Or you have entire blocks that actually are switching between the two haplotypes. So this is the most common situation in NGS assemblies. Here you start to have some phasing, but you still have large blocks that are switched between the two haplotypes. And this is the best possible scenario where you have completely separated maternal and paternal haplotypes into two distinct sequences. Here is also where you make the fewer number of mistakes overall, because whenever there is a difference between the two haplotypes, you always have the risk here at the boundaries of this sequence. You make a lot of mistakes because the overlaps are not clear. So this is also another reason why you want to, in principle, try to separate the haplotypes. This is it. I will finish with this slide. That's something we had planned, but in the end not implemented. Trying to run, instead of using Galaxy, trying to run this exercise on the cluster, the HPC cluster of the Rockefeller. Of course, it's much more complicated to install. But the idea is that James and Camila here, they put together a pipeline that can run on the cluster that takes care of basically what we have seen with Galaxy, and also more than that. So there are also other QC that are generated by the pipeline. As you can see, the principle is very similar to the one of designing a pipeline with Galaxy. With that, on my end, I'm...
One more thing I would like to point out, and how what Judo presented today show you that you need to look at assembly with a critical eye. If you see publication when people said, this is a chromosome level assembly, and they show you the scaffold in 50, that doesn't mean that is a very good genome, right? Look at the results to see what QC they did to support that is a good genome. Look for the contigent 50, look for the Busso score, look for a lot of things that we show you before. Usually you need to do more than just reading the paper to assess if it's a good genome or not, but you can sort out a lot of bad genomes when people claim it's a chromosome level genome.
Yeah, they will. I mean, if you read the paper, there will be a lot of selling in many of these papers, so don't trust them. But that's why we put so much emphasis on the QC part, because you don't want to use low quality genomes basically for your analysis, or even when they are high quality, you want to be able to try to understand if, let's say, a specific region you're looking at is actually assembled correctly or not, because if it's not assembled correctly, you may draw completely false conclusions based on what you see there. Could be when you're designing your probes, or when you are doing some evolutionary inference. But it's really important to keep that in mind.
And if a paper doesn't show you all the statistics we show you here, that's a red flag. I mean, that they don't meet some of the criteria that defines being a good genome or not.
Yeah, if they even haven't checked, it's very likely that it's not very good.
I've seen often some abstract of genomes where in the abstract they said scaffolding 15, that's it, right? And of course people are like, oh, that's cool, but no QV, no Busco, no IC plot, no config. So.
Is NG50 starting to be reported, or is that something, for example, I didn't search for it in Galaxy yet, but is that something that we're...
It's not very widely used. I don't like it. But...
Why don't you like it? So I understood that the NG50, that the benefit of it was that standardized it, right? So that you can compare different genomes, right? So that you sort of have a better indication of like, is this one better than the other?
It's standardized by genome size, essentially, but not on karyotype. That's what bothers me, right? So it's half standard.
So there are two problems. One is that it relies on your estimate of the real genome size, which may not be accurate. And the other thing is that, well, I would say it's better than the end. So Jean-Nicolas, just I don't know, Matthew and Zachary, if you're still with us, it's the idea is that, remember when we calculated the N50 and we said, okay, we have a 400 megabases. Now the whole length is 400 megabases. Then we do the half of that number and we count and here is just instead of using 400, which is the total size of the assembly, you use the expected genome size. The reason why this is much better is because in principle, if your assembly is very incomplete, you're still dividing for the total length and you get maybe a grid N50, but it doesn't show you that you have half the genome actually present. But if you divide by the real size, your N50 estimate is going to go down.
Yeah, and most of the genomes now are pretty much close to complete, like they're very high in completeness. So I don't see a big use of NG50, but that's my bias. I mean,
I would not use any of these.
Or when you try to compare two different species that are different number of chromosomes and different genome size, that's complicated, right? And NG50 will not address the distribution of your chromosomes. Right? See, one species has a larger number of few chromosomes, you get different N50 than species with large genomes and a lot of chromosomes.
So I personally, I think that the N50 gained a lot of momentum in the last 10 years or so, because long reads have this big advantage that tend to generate highly contiguous assemblies. So in order to sell, let's say, this idea of highly contiguous assemblies, N50 is really good because it's giving you immediately the sense that your assembly with long reads is much, much more contiguous than a short read assembly.
Yeah.
But when it comes to evaluation, it's tricky to
guess what PacBio does now? PacBio now said, oh, there is more than contiguity. You need to look at a QV. So of course, they always have a setting point. One for
yeah, exactly. Because the problem is that before introducing hi-fi reads, they had long context of high contiguity, but because of the higher error rate of long reads, they had low quality. So the consensus
so talk about quality.
QV was low, so they were never discussing that. They were just saying, "Oh, we have a great contiguity." Now that they can also claim that the actual sequence accuracy is very high, even higher than Illumina, then they start to advertise that again. But that's me.
But see, to go back to your question, I mean, Julia has a good point. It's hard to know a priori what is genome size, and that's a lot of guess. A lot of guesses. So UNG50 would be wrong anyway. So that's why I'm not very comfortable with it.
I mean, we've seen with Genome Scope that you can actually estimate it, but could be wrong. So, okay.